All right, everybody. Today we're looking at day 3 of distance learning, and we're going to talk about errors of meiosis and recap meiosis a little bit as well. So our learning objectives for today is we should be able to define what a karyotype is and how we use that in biology. And we're also going to talk about how you can differentiate between errors and meiosis and if they could potentially lead to an offspring being created or not. So just a quick meiosis recap. Remember, this only happens in sex cells. Again, we're going to get 4 genetically unique cells that are haploid. That means they have half of the genetic information or DNA needed to create an organism, and it's going to be random. So we know that those traits are going to move independent of one another. So let's look with karyotypes. So humans have 46 chromosomes or 23 pairs of chromosomes. Think about it like this. Pairs 1 through 22 is everything that makes up your body. The 23rd pair, however, is only for determining gender or sex biologically speaking. So if we're looking at the 23rd pair, if we see 2 X chromosomes, XX, we know that that individual is female. If we see an X and a Y, we know that that individual is going to be a male. So looking at this next karyotype, you can see that the chromosomes are organized from height, from largest to smallest. Again, chromosomes 1 through 22 are for bodies. Those are called autosomes. And then the 23rd pair in this case, we see there's one X and one Y, so we know that this individual is in fact a male. Again, just to reiterate that, we know, think of pairs as you can have one pair of shoes. That means you technically have 2 individual shoes. So 23 pairs of chromosomes in humans, 46 individual chromosomes that contain information. All right, so some possible mistakes that can occur in meiosis. We're going to look at this, and one of them is something called non-disjunction. It's basically where the chromosomes kind of stick together and they can't move apart of one another. This can happen in meiosis 1 or 2. Now, if this happens in meiosis 1, the effects of this are far more severe than if it were to happen in meiosis 2. And so if you look at that image there at the top half, you can see non-disjunction occurring in meiosis 1, and you can see that 2 of the gametes have more DNA needed, 2 of the gametes have less DNA than needed. If you look at the bottom half, you see that in meiosis 2, if that's when non-disjunction occurs, 2 of the gametes are actually viable and fine. The other 2, one has too much DNA and one has too little. And so those are the differences of where non-disjunction might occur and make some potential mistakes in meiosis. So again, if we have non-disjunction occurring in meiosis 1, it is far more severe because we're already starting the shuffling part, so to speak, not in the correct way. And so when we start to divide, quote unquote, the chromosomes, it's not going to be divided evenly. So I just wanted to give you a little bit reminder, and hopefully this visual image helps you get those two together. So what's the main point of this? Too much or too little DNA is bad, right? We don't want that. You need a certain amount of DNA because that's what codes for the instructions on how to make you, or how to make an organism a certain organism. So I want you to think for a minute, okay, great, we learned about non-disjunction. What's an actual case where this takes place in humans? And so I want to briefly give you a case study of what that looks like when this takes place in humans. So if we have non-disjunction occurring, one thing that we can have happen is something called Down syndrome. And so this is actually called trisomy 21. So what happens here is you get 3 chromosomes instead of 2 chromosomes of the 21st pair. So somewhere along the line, either in the egg development or sperm development, those homologous chromosomes didn't separate. And so now instead of having 2 number 21 chromosomes, we actually have 3. Okay. This karyotype is meant to just kind of give you a look at how this is. So I've boxed 21, so you can see that there's 3 chromosome 21s there. You can also see, hopefully, that by looking at this, you can determine the gender of this person. So take a minute, see if you can kind of determine the gender. You should have seen that this is a female because we see XX. Now, We've talked about non-disjunction and what that looks like if it's in autosomes, but you might ask yourself, what happens if we get a person that's XXX or XXY or only an X? And so that's kind of the lesson that we're going to look at next week, just to give you an idea about what we'll be talking then. Please make sure that you're asking any questions on that Google form, and I look forward to hearing from you, if only digitally. All right, this is Miss P signing